So hey guys, welcome back to my YouTube channel. Today again we are going to discuss a very short and precise topic from molecular biology, that is trans splicing. Splicing, as we discussed earlier, is basically the removal of introns, right? So uh, I hope the previous video was clear to you. Removal of introns. Now let's discuss this topic. Trans, what do you mean by trans? Trans means opposite or different. We say trans configuration in chemistry, we have studied, right? So trans means opposite or different. So here, let's see what is the definition of trans splicing. Exons from two separate RNA transcripts are spliced together to form a mature mRNA molecule. So here in earlier we have discussed that the source was single and formed two different products. Here there are two different sources. They combine to form one mature mRNA. Okay, so see, in trypanosomes, a single exon is spliced onto the 5' end of many different RNA transcripts produced by the cell. In this way, all the products of trans splicing have same 5' exon and different 3' exon. The reason for trans splicing is still unknown. So basically what they are trying to say is single exon spliced onto the 5' end. Means the 5' end of the exon remains same, but on the 3' end different types of exons are ligated, spliced and ligated. Now let's see here. In this we have this first one, that is exon from 5' end and intron, and this is pre-mRNA of different source, and this is spliced leader RNA from different source. So first we'll see this one. This comes down. This is exon. This is exon. This is intron. So this exon comes is spliced from here and ligated with the pre-mRNA one and forms a new one that undergoes trans-splicing. Basically, it is a splicing only. The fact is, there are different sources, two different mRNAs come and ligate together, and only thing you need to know is same 5' end of exon, but different 3' exons. Okay, so the you can see this. Then a Y-shaped structure is now removed out. That was the intron part. This is the intron part from the pre-mRNA. Okay, in all the, I have already told you 5' end remains the same, so it got removed on its own. So what is the chemistry of this reaction is same as that of standard splicing reaction. We have to note this point that the only difference in the formation of lariat in standard splicing, we have seen lariat formation structure somewhat like this, right? It is formed during splicing, and a Y-shaped structure, branch structure, is formed in transsplicing. We have seen this one, Y-shaped structure. This is because the initial reaction brings two RNA molecules rather than forming a loop within the single molecules. Here we have seen two two different molecules were brought together, and in standard splicing we see there is a single mRNA that undergoes lariat formation and then ligated, spliced and removed out. There are two types of transsplicing, intragenic and intergenic, right? Intragenic, intra means within. Okay, so intragenic means within the genes. Genic means genes. So two identical means two same pre-mRNA molecules from the same gene. From within the gene, there is a single gene. इसके बीच में से हमने दो mRNAs को निकाल लिया जो कि आइडेंटिकल है, फिर उनको स्प्लाइस आउट करके लाइगेट कर लिया हमने, राइट? This is introgenic. Are spliced together to generate a mRNA with duplicated exon sequences. And intergenic means among two different genes generated from two pre-RNAs, pre-RNAs derived from different genes. Basically, one gene from this and one from this ligate together to form. Okay, hope you understood the topic. Please like, share, and subscribe the channel. Thank you.